Hello everyone. The today's topic of our discussion is that the cellular mechanics of the sodium to cause hypertension. कि cellular level पे sodium किस तरह hypertension करता है. Okay. Now come towards the first mechanism. First mechanism is that sodium, as we know, Na is osmotically active. So it means that where the sodium will go, water always follow the sodium. So it increases blood volume. So when blood volume will be increased, indirectly the total peripheral resistance will be increased. Increase total peripheral resistance. So when total peripheral resistance will be increased, uh, that will leads to the hypertension. So this is the first mechanism that how sodium is going to cause the hypertension. Now come towards the second mechanism. So to understand the cellular mechanics, that how sodium at the cellular level going to cause hypertension. So first of all we have to little bit know about the contraction mechanism of a blood vessel. Okay, for example, this is a blood vessel, and we all know there are different type of receptors are present on the surface of the blood vessels. And the receptor, the name that I am going to write now is G protein, G protein 1230. Okay, and this G protein is associated definitely with its receptor, which is called as G protein coupled receptor. So this is a G protein coupled receptors, and it is associated with a G protein here. This G protein, which is basically G protein 12 to 13, and it consists of three subunits: alpha, beta, and gamma. Okay, and this G protein 12 to 13, ये जो G protein 12 और 13 है, ये inhibitory in nature है. It is inhibitory in nature. Inhibitory in nature. It means that it is associated, or it is GI protein. This G protein 12 to 13 is GI protein. GI okay. So on the surface of the blood vessel there is an other effector protein that is called as adenylyl cyclase. So this adenylyl cyclase increases the cyclic AMP level, and this cyclic AMP level increases the level of protein kinase A. Protein kinase A. And this protein kinase A causes the uh, in nature in normally protein kinase A causes the phosphorylation of a protein or an enzyme which is protein in nature is called as MLCK, means myosin light chain kinase. Myosin light chain kinase is an enzyme. So protein kinase A causes the phosphorylation of this protein. When the phosphorylation of the myosin light chain kinase happens, it gets deactivated. There are in normal cases when any protein gets phosphorylated, it becomes active. But in case of myosin light chain kinase, it is from one of those protein. ये उन प्रोटीन में से है जब इसकी फॉस्फोराइलेशन होती है तो ये इनएक्टिवेट हो जाती है. ओके. So this myosin light chain kinase then causes the phosphorylation of myosin head, which is involved in the contraction of the blood vessel. This is myosin head. Myosin head. So myosin light chain causes the phosphorylation of myosin head, and contraction of the blood vessel take place. Contraction of blood vessel. Contraction of blood vessel. So in this way contraction of the blood vessel take place. Okay, what sodium is going to do with this cellular mechanic? Sodium, which we basically use normally, this sodium activates this GI protein. Okay, when this GI protein will be activated, the activity of the adenylyl cyclase will be decreased, because we know G protein, which is 12 to 13, is inhibitory in nature. Adenylyl cyclase activity will be decreased, so cyclic AMP level will be decreased, and cyclic AMP level will be decreased, protein kinase activity will be inhibited, and when protein kinase A activity will be inhibited, the myosin light chain kinase will not be phosphorylated, and we all know when the myosin light chain kinase gets dephosphorylated dephosphorylated it gets activated dephosphorylation phosphorylation and it gets activated because protein kinase activity has been reduced because of this second messenger pathway. Myosin light chain kinase will be activated, and myosin light chain kinase will be activated. It will be uh, causing the phosphorylation of the myosin head, and in this way the contraction will be increased. When we use a lot of sodium and unusual increase in the sodium intake, this mechanism remains active for the longer period of time. जब ये ज़्यादा टाइम के लिए एक्टिवेट रहता है तो मायोसिन लाइट चेन काइनेज एक्टिविटी इन्हांस हो जाती है. तो मायोसिन लाइट चेन काइनेज एक्टिविटी इन्हांस हो जाती है तो मायोसिन हेड की फॉस्फोराइलेशन के चांसेस भी बढ़ जाते हैं. तो इस तरह ब्लड वेसल ज़्यादा देर के लिए कॉन्ट्रैक्ट रहती हैं. दैट लीड्स टू द हाइपरटेंशन. हाइपरटेंशन. Okay, this is the first mechanism at the cellular level. Okay, now come towards the another mechanism that is associated with a receptor system, and the name of that receptor system is rho-kinase or H-O-kinase system. So rho-kinase system is present on the surface, or RHO receptor are present on surface of the blood vessels, and these receptors are associated with second messenger as well. Second messenger. Okay. And because of those second messengers, those are associated with rho-kinase, causes the activation of uh, an enzyme that is called as MLCK, myosin light chain phosphatase. Okay, so this is an enzyme. This is myosin light chain phosphatase. Okay, and what is the function of this myosin light chain phosphatase? This myosin light chain phosphatase basically causes the dephosphorylation of this myosin head, means this myosin light chain phosphatase removes the phosphate from the myosin head, or in this way myosin light chain phosphatase uh, decreases the contraction phenomena. Okay, but when we use unusual increase in the sodium, okay, so sodium activates this rho receptor, and this rho receptor when associated with second messenger system then activates this second messenger, inhibit this myosin light chain phosphatase. Listen it again. The sodium activates the Rho receptor. Rho receptors, when activated, it activates its second messengers, just like this G protein, GI, adenylyl cyclic AMP. Okay. So when those second messenger gets activated, myosin light chain phosphatase become inhibited. Okay. When myosin light chain phosphatase will be inhibited, there will be the no dephosphorylation of this myosin head, and there will be the uncontrolled myosin light chain kinase associated phosphorylation of the myosin head. So in this way, by these two interconnected mechanism, sodium increases the blood pressure. So definitely when myosin light chain phosphatase will be inhibited, it will not be able to cause the dephosphorylation of this myosin head. When myosin head dephosphorylation will not be happened, blood vessel will be contracted for the longer period, and that will lead to the hypertension. So this is the third mechanism, that how sodium is going to cause hypertension. Okay, now come towards the fourth or the last mechanism. So just like those mechanics that we have studied right now, this is blood vessel. Okay, this is blood vessel. Okay, on the surface of blood vessel, there is a transporter or pump. This is pump. Okay, and the name of this pump is sodium potassium pump. Sodium potassium pump. Okay, and what is the function of this sodium potassium pump? This sodium potassium pump is associated with the outflux of sodium and influx of potassium ions. Okay, and on the blood vessel, this is basically the blood vessel. Blood vessel. So on the same cellular surface of the blood vessel, there is an other transporter that is called as sodium calcium exchanger. Sodium calcium exchanger. Okay, so what is the normal behavior of this sodium calcium exchanger? It causes the inward or influx of sodium and outflux of potassium ions. Sorry, calcium ions. Okay, in this way, these two pump and calcium exchanger work with each other. Okay, so this is the blood vessel. Now come towards the main character, which is sodium right now for us. Sodium. Okay, so when we use sodium in excess or unusual increase, or we can say if we uh, use the sodium for longer period of time in excess amount. This is most appropriate word. The use of sodium for longer period of time in excess. Okay, so this is the main characteristic of sodium. So when the sodium is in excess, it causes the release of a compound in the human body that is called as ubain. Uabain, ubain. It is a compound which has its property just like glycosides, cardiac active glycosides. Okay, so this is a compound ubain. Okay, ubain has been released, or excess of ubain has been produced in our body because of uh, excessive use of sodium. So now this ubain will come and will be going to inhibit this uh, sodium calcium exchanger. So this sodium calcium exchanger will be blocked. Okay, so when this sodium calcium exchanger will be blocked, means that the retention of the sodium inside the cell will be enhanced, because this pump was responsible for the outflux of the sodium. When it is blocked, the sodium will not be able to go outside, or the we can say sodium influx will be ceased. Okay, so intracellular sodium activity will be enhanced. So when sodium activity inside will be enhanced, this exchanger activity will automatically be decreased. Automatically, automatically decreased. Why? Because the cell sense that the sodium is already in an excess because of the blockage of the sodium pump. So this exchanger will be indirectly blocked by the sensing of increase in the sodium. Okay, so in this way the calcium outflux from the blood vessel will be decreased. So in this way intracellular calcium will be enhanced. Okay, when in the blood vessel the calcium is in excess, it will bind with a protein that is called as calmodulin. 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 Okay, so this calmodulin then causes the dephosphorylation dephosphorylation of MLCK that we have just studied in the previous slide. MLCK, MLCK. Okay, as we know, when the dephosphorylation of the MLCK happens, it becomes activated. Yes, activated. So when it becomes activated, it causes the phosphorylation of myosin head that we have already just studied in the previous slide. Myosin head. So when the myosin head will be permanently will be phosphorylated by the myosin light chain kinase because of its dephosphorylation associated with the calcium calmadulin complex. When the calcium binds with the calmadulin, it is called as calcium calmadulin complex. So this calcium calmadulin complex causes the dephosphorylation of myosin light chain kinase. When myosin light chain kinase will be activated, it will cause the phosphorylation of the myosin head. The myosin head will cause the contraction of blood vessel, contraction of blood vessels, and that leads to the hypertension. Hypertension. Okay. So this is the fourth cellular mechanism that how persistent or prolonged use of sodium in excess leads to the hypertension. Let's have a quick recap. So when we use excessive sodium, it releases the ubiane, which is a compound releases in our body, and it has the property just like cardiac glycosides. Okay, so this when inhibits this potassium sodium pump. When this sodium potassium pump will be blocked, it will increases the sodium, and this increase in the intracellular sodium is associated with the blockage of this sodium calcium exchange activity. So this sodium calcium exchange activity inhibition leads to the increase of the calcium inside the cell. So when the calcium inside the cell increases, it causes the complex formation with the calmadulin. This calmadulin activates myosin light chain kinase. This myosin light chain kinase then activate the or when it is activated, because of the phosphorylation of the myosin head persistent phosphorylation leads to the or outcome in contraction of the blood vessel for the longer period of time, and in this way hypertension happens. So these were the four cellular mechanism that how sodium is going to cause hypertension. If you enjoy this lecture, please like, subscribe, and share.